if you would, and it could be supplied. I mean, from the point of view of nutrients, supplements, and nutritions through the synovial fluids rather than the vascularization by the blood vessels. This is actually because the cartilage is going to be part of different supporting and flexible at the same time structures. So, as a component, we have also protein components and non-protein components in the cartilage, and mainly we have collagen type 2 as a distinguishing protein part in the cartilage, representing more than 90% of the proteins that we have in the cartilage, as well as the presence of other types of cartilages, and they actually play a role in the different structures of the cartilage types because we have different types of cartilage. For example, we have elastic one as well as the fibro elastic one. So these types of collagens are playing a role in the elasticity of the cartilage tissues. For example, we have type 5, 6, 9, 10, 11, for example. They are also present in order to support collagen type 2 or to make connection between the collagen and other types of proteins. As an example here, just to mention that we have what's called elastic cartilage, which contains elastin besides the collagen, and it's different type of protein, while the fibroelastic cartilage contains type 1 collagen as well in order to have more fibrous structure. As I mentioned before, cartilage is a vascular tissue. It means that the blood vessels are not going through the tissue itself, and it will get its nutrients from the surrounding fluid and is known as synovial fluids. Can be destroyed as a matter of physiological process or pathological process by certain proteases, and specifically they are known as collagenases, as well as the action of stromalysin enzyme that has the same function of the collagenases in order to destroy the collagen. So the collagen can be degraded or synthesized according to the specific type of that tissue, or it can be pathological issue due to the stimulation of certain pathological pathways. These proteases have been found to be activated by the action of certain cytokines. It means that once we have inflammation, we expect the destruction of the cartilage tissues due to the action of these cytokines and interleukins, such as interleukin 1 and TNF alpha. It has been found that the TGF, which is known as transforming growth factor, as well as insulin-like growth factor, playing a role in the cartilage anabolism. So just to mention the importance of the different triggering or stimulating molecules like the cytokines or growth factor markers like the TGF and insulin, IGF, for example, one that can play a role in the metabolism and the structure of the collagen. I mean, the cartilage itself. Now, as we have non-collagen proteins in the bone, we have also non-collagen proteins in the cartilage, but with some differences compared to the bone structure. So we have proteoglycans, like the ones that we have in the bone, as well as the presence of another protein, which is called aggrecan. The aggrecan is playing an important role in the cartilage structure because it is one of the major components that makes the cartilage looks like a jelly structure compared to the bone, okay, rather than being compact. As well as the presence of other proteoglycans that we have seen in the bone. They are also present here because they have to support the structure of the collagen, as well as the chondronectin, the thing that we didn't see in the bone. We can see it here in the cartilage, and it can play a role in the binding. I'm sure the laser just to okay. Okay. Uh, this is the rule of the chondronictin protein, which is the connection or the linkage of collagen type 2, for example, and anchorin C type 2, which is also important for the anchorage of the collagen type 2 on the surface of the cartilage cells واللي هم عارفين باسم الchondrocytes. So these are the basic components as protein components of the cartilage tissue compared to the bone structure. Talking about the proteoglycans that we have in the cartilage tissue and how they make the connection between the fibrous collagen type اللي هو type 2 collagen, and this is the fiber of the collagen as well as the other fiber here. They are connected together with what's called proteoglycan, which is similar to the proteoglycans that we have seen earlier in the bone. And if we take cross section in this area, we can see the presence of the core protein as well as the chondroitin sulfate اللي هو carbohydrate molecules that are integrated in the collagen type 2 in order to have the complete structure here. And this is the hyaluronic acid that is connecting the carbohydrate molecule with the protein. The difference here between the collagen and between the cartilage and the bone structure is the presence of this molecule, which is known as aggrecan. And the aggrecan molecule actually is coded by a gene that has repeated sequences. لما بحكي repeated sequences, for example, we have ACGTA, for example, and this sequence is going to be repeated many times. They are called tandem repeats. So in this gene specifically, اللي هو gene الأجريكان, we have repeated sequences that can, and these repeated sequences, by the way, they are coded, which means that الأجريكان can be longer or shorter, okay? And the length of the aggrecan itself, which is connected with the hyaluronic acid, for example, in this case, through the connection of linker protein. So the aggrecan is playing a role in the texture of the collagen. And nowadays, we are studying here at Yarmouk University the relationship between the length of the aggrecan of repeated sequences, يعني قديش طول الأجريكان, for example, and the occurrence of certain disc degenerative disorders اللي هم الاختلالات أو الاعتلالات اللي بتصير بالديسك عند بعض الناس. So it can be or it could be genetically predisposed disorder. It means that إنه في ناس عندهم genetic predisposition اللي هو الاستعداد الوراثي إن يكون في عنده مشاكل في الديسك مقارنة بالأشخاص اللي ما عندهم هاي الريبيت. بمعنى آخر length or structure للأجريكان could play a role in this story of disc degenerative disorders. So this example here is provided by bovine nasal cartilage as an aggrecan structure, which represents the core protein, as you can see here. Okay, and here we can see the extensions of the chondroitin sulfate, اللي هم carbohydrate molecules, as you can see. And we have also connection points of other things that could connected with the aggrecan molecule. فالأجريكان بمعنى آخر making a kind of network connection between the proteins that we have in the cartilage as well in order to have a well-formed structure. As a matter of disorders that can be associated with the cartilage structure, some individuals could develop what's called chondrodysplasias disorders that can be associated with type 2 collagen mutations, for example, or FGFR mutations, which means the following. هلا الكولاجين type 2 is a structural protein. Structural protein فيه mutation نقدر نفهم إنه إذا صار في مشكلة بالstructure we could have abnormal chondrocyte structure as well as the development of what's called chondrodysplasia اللي هو abnormal growth of the or abnormal structure of the cartilage. While the FGFR is abbreviation of what's known as fibroblasts growth factor receptor, and specifically type 3, is metabolic pathway. بمعنى آخر بالأول كان في عندي مشكلة بالستركتشر، الآن مشكلة بالريسبتور الموجود على هذول الخلايا، ففي مشكلة عندي بالسيجنالينج باثوي المسؤولة عن الديفلوبمنت أو الفورميشن أوف ذا فايبروبلاست. اوكي. as well إنه في عندنا الكولاجين type 2 we have also some minor other proteins that can be mutated in order to have chondrodysplasia, but mainly the main reason is the or the abnormal structure of collagen type 2 in the cartilage structure. So any mutation in this type of collagen, especially the alpha 1 chain, could develop what's called chondrodysplasia disorder. As an example, we have what's called Stickler syndrome, which is manifested as a condition or as a phenotype by degeneration of joint cartilage and the vitreous body of the eye. So this kind of disorder is going to affect the joint as well as, or joints in general, as well as what's called vitreous body of the eye, اللي هو التجويف الداخلي للعين أو الغشاء الداخلي لتجويف العين. That can be affected by this kind of disorder if we have a mutation in collagen type 2A1. As a common example, the best known chondrodysplasia disorder is the disease known as achondrodysplasia or achondroplasia, which is associated as a manifestation with what's called short-limbed dwarfism. It's kind of dwarfism that is related to the short limb phenotype. بمعنى آخر إنه أرجلهم قصيرة هذول الناس. As a very famous example, Michael Ian is a pediatric surgeon, orthopedic surgeon actually, at Johns Hopkins Hospital, one of the best medical institutes in the world. He got that position at Johns Hopkins Hospital after 20 letters of rejections from medical schools due to his condition. And this kind of condition doesn't mean that this patient is, or this individual actually, is mentally retarded. He just have or has that kind of condition, which is called achondroplasia, due to the mutation in the gene that is responsible for the formation of the cartilage. And as we talked about this story earlier لما حكينا عن ممكن تكون مشكلة بالكولاجين، ممكن تكون مشكلة بالFGFR receptor, fibroblast growth factor receptor type 3. If we have such kind of mutation, we will have replacement of certain amino acid, and eventually we will have abnormal development of growth of the cartilage due to إنه هذا الريسبتور mutated. لانه كنا تكلمنا سابقا بالاندوكراين سيستم انه حتى الخليه تعمل وظائف تبعتها، واللي هي على سبيل المثال وظيفه الديفلوبمنت of the structure. We have to have certain triggering molecules. So if the fibroblast growth factor اجى على هذا الريسبتور والريسبتور هذا مش شغال لانه في mutation, we will have abnormal formation of these cells, and it's associated with what's called achondroplasia condition. And this is the summary of the chapter. These are the most important things that you have to focus on when you study this chapter or this slide, in fact. Now, moving to the next topic in our story, which is the discussion of certain markers that we could use in the diagnosis of bone or cartilage disorders. So when we say markers, it means that we are going to make a diagnosis depending on the presence of these enzymes, and specifically the activity of these enzymes or the presence of certain molecules in the circulatory bloodstream. In a simple way, نحكيها بالعربي، لما نعمل diagnosis بالعادة أمامنا الخيار المباشر اللي هو الكلينيكال assessment، وبعد الكلينيكال assessment we can make what's called ruling out of the certain disorders. يعني بدي أعمل أنا confirmation of my case أو rule out of my case. وبالتالي باخذ عينة blood، وهي عينة البلد ممكن أخذ منها أنا serum for example، وأبلش أعمل analysis للسيرم هذا، وهون ببلش بشيء اسمه clinical biochemistry approach. انه بدي اشوف الاشياء اللي موجوده في هذا السيرم. ممكن تكون جلوكوز اذا بدنا نفحص دايابيتس، ممكن تكون any other type of enzyme activity اذا بدي اشوف في خلل في تيشو معين. And we could search for certain markers like molecules بعض الجزيئات or certain ions. هلا هذا كله داخل تحت الامبريلا ما يسمى او ما يعرف باسم الكلينيكال بايوكيمستري. هلا the other option could be the genetic investigation اللي هو المولكيولار investigation or molecular diagnosis. بنروح على الجينات وبالتالي بنعمل extraction للدي ان اي من هذا الشخص، او ممكن يكون الار ان اي depending on the approach, and we could analyze the DNA sequence to figure out if we have mutation in that gene or not. So we have a specific approach to go through, but most probably you are going to extract the blood samples from the individual in order to rule out or confirm the condition. So the things that we are going to talk about as disorders that can be investigated by certain markers through the blood samples. Duchenne muscular dystrophy, for example, and Duchenne muscular dystrophy, in fact, can be diagnosed by clinical assessment from the phenotype. Glycogen storage diseases that are related to muscles, because the glycogen storage disease are almost associated with the liver, as well as the role of the mitochondrial diseases in the muscle disorders. بمعنى آخر إنه إذا في عندنا مشكلة بالmitochondrial function, most probably we have problem in the nervous system as well as the muscular system. ولما بحكي muscular system, it could be any other type of muscles, cardiac or skeletal muscles. And we will talk about osteogenesis imperfecta again, and we will add what's called Ehlers Danlos syndrome, اللي هي stretchy skin syndrome. As we learned before, no, the bone is a dynamic structure. We have mineralization process or formation process, as well as resorption process of the bone that rely on the presence of osteoblast as well as osteoclast cells, and this is normal process. So if we have imbalance in these pathways, the patient or the individual could develop certain disorders that are related to these mechanisms. Okay, and we can see the markers that will be associated with these conditions. بمعنى آخر، يعني إذا كان في عندي هون على سبيل المثال resorption أعلى من formation, I could see certain markers that are related to the structure of the bone. يعني خلايا البون عمالها بتتكسر وبتكسر البروتينات اللي حولها وبعطونا مجموعة من الفراجمنتس اللي المفروض كانت بشكل طبيعي والآن fragmented. So after fragmentation, they will appear in the blood, and we can detect these markers in certain techniques. So what we are going to test could be enzymes in the blood, or could be non-enzymatic peptides or molecules. So you are going to check the presence of these markers in the blood in order to have clinical assessment in a precise way. So the markers that are related to bone resorption are due to, they are present in the blood due to the collagen breakdown of products such as hydroxyproline. We could find it in the blood because we have resorption. طيب انت قاعد تحكي لي هون انه breakdown of collagen products. وشو علاقتها بالresorption? Resorption means that you are forming a bone. So as you are forming a bone, it means that you lost some of that bone due to any type of condition. Could be trauma, could be fracture, could be anything, or inflammation, for example, and you are trying to repair that. So as a marker of bone resorption, you could see such fragments of collagen types, or you will see, for example, high activity of certain enzymes like alkaline phosphatase as an enzyme. And in this case, we have what's called bone CL protein, اللي هو acidic phosphoprotein, that is responsible for the nucleation or mineralization process in the bone. So you can see the activity of these enzymes or the presence of these markers in the blood stream. For example, you can see also cathepsin K as another marker. Another example of the bone formation is the alkaline phosphatase, which is responsible for the building up of the bone. Because if we have higher activity of this enzyme, it means that uh, there is something wrong with the bone. So it means we have problem with the tissues that are producing this enzyme or that are rich with such kind of enzyme. So osteocalcin is another example of the bone formation. And here we have general comparison between bone formation markers as well as bone resorption markers. And this is the acid phosphatase protein that is related to the bone. It's called bone acid phosphatase or acidic phosphatic acid protein, as well as collagen type 1 that can be detected in the bloodstream due to the formation of the bone. Osteocalcin as another marker, and these are the osteoblasts, of course. When we have resorption of the bone, it means that the bone is going to be deformed due to the action of maybe osteoclast cells. You will see these fragments of proteins that will be generated from the breakdown of the bone structure. So these markers are just the fragments of the proteins that we could have in the blood due to the destruction of the bone. The calcium is another indication as an ion, as well as a marker for the bone resorption in the bloodstream. So bone formation and bone resorption markers, it means that you could have the same marker for both processes, because if you have bone resorption, it means that you are going to replace this bone with the new contents of the bone. بما انه ان عمالك بتكسر معناته ان عمالك بتبني بداله جديد. او بمعنى اخر, if you activate resorption, sometimes you could activate formation of the or deposition of the bone itself. So we have some kind of overlapping markers خلينا نسميهم اللي هو الهيدروكسيبرولين can be found when we have bone resorption or bone formation, as well as الOC fragments اللي هو الفراجمنتس اللي طالعين من الاوستيو كالسين بروتين. عمر بني مرعي اتفضل.
دكتور بصير بسترجع على الرسائل قبله.
يس.
بالفورميشن في كولاجين تايب 1 بروبيبتايدز از ماركر, وفي بالريزوربشن في كولاجين تايب 1 تيروبيبتايدز. ايش الفرق بينهم؟
التيروبيبتايدز اللي هم الفراجمنتد U2 degradation هذول هسه بنشوف تبعهم بنسميهم تيلوبيبتايدز لانه جايين على الاطراف. تيلو معناها at the end of something, على الطرف من هون من اوكي. هلا البروبيبتايدز هذول اللي هم الالفا تشينز
اللي حكينا عنهم بالمحاضره الاولى,
اللي هم بكونوا الكولاجين في البدايه.
فهمت عليك.
هلا اوريدي هذول مكونين ولكن they are fragmented due to the destruction. هسه قلنا الاوستيوكالسين يعني واحد من الماركرز اللي عليه اشكاليه اللي هو البون فورميشن او ريسوربشن. هذا ببين بعدين بالكلينيكال اسسمنت يعني تو كونفيرم ذات اور نوت. هلا التيبل اللي هون بيلخص هاي الاشياء وبيلخص الطرق اللي ممكن نعمل فيها الاناليسيس والطريقه اللي بناخذ فيها السامبل. What I want you to know or to do is smart studying. تمام؟ بمعنى هلا هذا التيبل بيحكي عن ماركرز اوف بون تيرن اوفر بشكل عام كل التيبل, وبالبدايه لانه في اكثر من جزء من هذا التيبل خلينا نورجيك اياهم. وبالبدايه بيحكي عن الماركرز اوف بون فورميشن. تمام؟ هلا بعض الماركرز هذول could be treated as bone specific markers most probably, على الرغم انه موجودين في اماكن اخرى, ولكن هذول نعتبرهم احنا most probably اللي هم البون ماركرز like الاسيد فوسفاتيز as well as اللي هو bone acid phosphatase and او acidic protein والalkaline phosphatase enzyme. So these proteins or enzymes are present in the bone, so that they are bone specific most probably, unless we have other conditions like cancers in certain other tissues. And the sample always is the serum sample most of the time. So you could take a blood, and we have these immunoassay protocols. هلا الاميونواساي قصتها يا جماعه بشكل بسيط جدا, لان انا وعدت احكي عنها وخليني احكيها اليوم مشان نبرئ ذمتنا. الاميونواساي بتقوم على مبدا بسيط جدا انه you have you have ليش اوكي هون ممكن طيب you have this بروتين وخليني اسميه انا انتيجين. هلا بسميه انتيجين لما بكون في عندنا antibody for example that can bind this kind of antigen, okay, and this antibody is conjugated with enzyme. هاي الميثود موجودة باسم إلايزا. تمام، يعني من أسهل الطرق للإميونو أساي ديتكشن اللي هي كلمة إلايزا. هلا بشوف مثلا راديو إميونو أساي the same، يعني basic approach إنه في أنتي بادي ولكن الماركر بيختلف. هلا من أسهل الطرق اللي بنستخدمها very common بالأساي اللي هو الإلايزا. مبدأ الإلايزا بيقوم على إنه إذا في عندك الأنتيجين موجود، إذا القطعة اللي بتدور عليها الماركر موجود، الأنتي بادي راح يرتبط فيها، and then because of the presence of the enzyme you will add substrate which is almost colorless and you will get colored product. بمعنى آخر لما بتحط السبستريت على الرياكشن، تمام، انت بتاخذ عينة بلاد فور اكزامبل تحطها في ديفايس معين هو عبارة عن سمول ويل، يعني عبارة عن حفرة صغيرة من البلاستيك تكون على شكل بليت، and you will add the antibody، تمام، then you will add the substrate. So it's kind of chemical reaction you can do in the lab. If the color is changed, for example, to be yellow, blue, whatever the kind of the product, I mean, if the color is changed because sometimes إنه السبستريت بيكون ملون بعدين بصير colorless, it means that إنه الإنزيم موجود، مما يدل على إنه الأنتيبادي ارتبط بالأنتيجين، مما يدل على إنه الأنتيجين موجود. In a simple way الإلايزا والإميونو أساي كلهم بيعتمدوا على هذا المبدأ، إنه antibody binds the antigen and then the substrate will be converted to the product so you will see a color. هلا الانتنسيتي اوف ذا كلر، شو يعني انتنسيتي؟ شده اللون، كل ما كان اكثر دكونه خلينا نسميها more dark it means that انه الكونسنتريشن لهذا الانتيجين is higher. Okay, so in the lab we have specific protocol to go through اللي هو لازم يكون في عندي standard عشان اقارن فيه واقول انه انا في عندي مثلا high concentration of this antigen or low concentration of this antigen. Okay, we can use for example electrophoresis or we can use enzyme assay. كثير من الميثودز اللي انا بتمنى بيوم من الايام انه إن نقدر اشوفها انا وياك وان شاء الله نرجع الجامعه في احدى المختبرات للاسيز اللي بنعملها. الان فكل الطرق تعتمد بالاساس على الاميونو اسي او الاكتيفيتي. الاوستيوكالسين as well is can be found in the bone or platelets, so it's not highly specific to the bone, but we can make primary assessment, clinical assessment, and we have here what's called C-terminal propeptide of type 1 procollagen and N-terminal propeptide of type 1 collagen، لأنه type 1 بالبون، وبالتالي يمكن تشوف أنت الطرف اليمين أو الطرف اليسار لهذول الفراجمنتس اللي موجودين بسبب إنه في عندنا bone formation due to certain condition. أنت بتكون البون لأنه ممكن يكون كان في fracture or we could have sarcoma اللي هو tumor. these markers are associated with the building of the bone. هلا ممكن بعض الأحيان يكون physiological بدناش نخربط. هلا الأطفال بتكون عندهم الكالين فوسفاتيز أعلى لأنه في عندهم growth. البريجننت women راح يكون عندهم الكالين فوسفاتيز أعلى لأنه they are pregnant and they have infant that is developing bone. So بدناش نخلط بين physiological و pathological conditions. We have to be careful. هلا هدول الأشياء اللي هو مرتبطة بالريماركس تبعونهم. I'm not going to do clinical assessment now مش مطلوب منكم, but as an advice please keep this table with you لما تروح السنة الرابعة والخامسة والسادسة بالكلينيكال تكون هاي الأشياء موجودة معكم as a guideline for the clinical studies.
طب دكتور،
yes
بهذا الفصل ايش مطلوب منا بهذا الجدول؟
أيوه حكيت من قبل شوي.
okay. يعني أول 3 عواميد.
هلا كل الأسي بشكل عام بيعتمد على الإميونو أسي. كلمة إميونو بغض النظر إلايزا ولا راديو إميونو أسي ولا بريسبتيشن كله إميونو أسي، بمعنى إنه في عندي أنتي بادي. تمام، لأنه أنا براعي إنه أنت عمرك ما شفت هاي التكنيك ولا اشتغلتها بإيدك، يعني you just heard about it. هلا another condition could be اللي هو bone resorption, and we have these kind of markers hydroxyproline that will be found in the blood as well as hydroxylysine glycosides, and most probably you will see them in the urine rather than in the serum. You could find them in the serum as well, and the assay could rely on the immunoassay or other specific techniques like HPLC. Okay, so just to know the difference between the bone formation and bone resorption markers, and here we have longer list of bone resorption markers اللي هو البيريدونيلين for example deoxypyridinolene اللي هو deoxyform of the pyridinolene. هلا ليش بنضطر نستخدم اكثر من ماركر؟ لانه بنشوف الكونديشن تبعتنا السيفيريتي تبعتها، انه الديستركشن هذا تبع البون جاي من أي condition for example. And most probably we will see degradation of collagen type 1. أنا بديش تفاصيلهم just to know that we have these types of collagens. الشيء المهم جدا إنه هذا الموضوع مش مرتبط فقط في البون. It could be associated with other types of tissue like the dental tissue. هلا في other proteins بس مش collagenous يعني مش collagen origin. Okay. And can be sometimes bone specific like osteocalcin fragments. You can see them in the bone, in the urine or in the blood as markers of the bone resorption due to degradation or bone synthesis, bone formation. The bone CL protein as well. And we can see these enzymes. هلا الفرق بين هذول وهذول إنه هذول عبارة عن proteins وهذول عبارة عن enzymes. Okay. اللي هو TRAS اللي بدك تسميه أو TRAC B tartarate resistant acid phosphatase enzyme is another marker. وللأمانة خليني أكون صادق معكم هذول الماركرز very special markers. يعني واحد من الجنرال ماركرز الروتين ماركرز بنستخدمهم بالمختبر احنا الالكالين فوسفاتيز والاسيد فوسفاتيز. اوكي we can go further اذا كان في عندنا يعني specific condition that we are digging to rule out or to confirm the condition. So in general, I wanted to summarize what we talked about and the most important things that are related to these markers. For example, alkaline phosphatase enzyme. Okay, the specification of that enzyme is a marker of bone formation. The osteocalcin as well. Procollagen type 1 prototype as another marker. The resorption is associated with hydroxyproline, hydroxylysine, glycosides. And here is the answer of the مين يسألني ما بعرف مش متذكر الاسم يعني للأسف بس here we can see what's going to happen to the collagen structure if we have destruction of that collagen. So we could have this fragment اللي هو C-terminal fragment تطلع لوحدها or the N-terminal fragment اللي اسمهم pyridinium cross-linked markers PYD PDP ممكن تسمعهم في المستقبل إذا اختصيت العظام إنه هذول markers جايين من collagen type 1 degradation as markers of the bone resorption for example. Okay, and they originate from collagen type 1. And we have also this kind of fragment NTX for example, and we have NTX assay for this fragment specifically. هلا كل قطعة من هذول أخذت فحص خاص لأنه الأنتيبودي اللي بيرتبط فيها مختلف. And we have CTX MMP اللي هو نتيجة الميتال بروتيازيس enzymes لأنهم بيشتغلوا على هذا القطعة أو هاي الفراجمنت. Okay, and we have CTX assay for example for the octapeptide here that can be produced by such kind of degradation procedure. So PYD again هي القطعة اللي جاية من fragmentation of these ends. So we have PYD and PDP حتى ما نقول إنه مش عارفين من وين هي جاية. So it's fragment of collagen type 1. Okay, هلا التيلوبيبتايد the same thing اللي هو fragments من C-terminal أو N-terminal بنسميهم إحنا telepeptides that will be generated through the degradation of the collagen. ممكن هذا degradation جاي من enzyme activity تكون genetic ممكن تكون من inflammation activation of immune system autoimmune system that will stimulate the degradation enzymes. Since we are talking about muscular system as well, let's talk about a very dangerous disease because from emotional point of view, this disease is kind of يعني socially requires certain kind of attention as well as healthcare even at home. لما حكيت emotional بسبب بسيط جدا إنه هذا disease is progressive disease and I think you have heard about this. وأنا حبيت أحط هذا السلايد حتى أشير لكم إنه you could find a textbook and as long as I remember it was the 4th edition talking about Duchenne muscular dystrophy. Duchenne muscular dystrophy due to the deficiency in the dystrophin protein that is required for the muscle structure or muscular tissue structure. So this disease again is progressive and it's genetic disorder that is characterized by progressive degeneration of muscles وبتبلش تشوف المريض عماله بنهار قدامك سنة ورا سنة ولهذا السبب أنا حكيت إنه emotionally it's bad disease. when you see someone that you cannot offer any help because it's genetic, and hopefully we can develop certain kind of genetic intervention to treat such kind of patients. So we have different types of muscular dystrophy or Duchenne muscular dystrophy, in fact, and they caused by the insufficiency of the dystrophin protein. هذا البروتين معمل بنصنع بطريقة صحيحة بالنسبة لهدول الناس, and the onset is in the early ages, and X-linked disease, in fact, the prevalence is very high in the male infants because it's X-linked disease. And when you say 1 to like 4,000 male will suffer from this kind of disease, it's high prevalence of genetic disorders. And just to know what we are talking about, this is the dystrophin structure protein that is required for the integrity of the muscle tissues. So when we have such kind of deficient condition of dystrophin, the actin proteins cannot make the right connection with the muscle cells in order to have the skeletal muscle structure. And as a phenotype or manifestations of the disease, this paper is describing such kind of condition. It's just a case report of an individual with Duchenne muscular dystrophy. He is 15 years old, and you can see the deformity of the body itself due to the destruction of the muscles because of the deficiency of such kind of dystrophin protein. That's why it's called Duchenne muscular dystrophy disease. And in another publication that has been found in Saudi Arabia, we can see just to have an idea about what's called molecular diagnosis. بنحكي ايش يعني molecular diagnosis. For example, هلا انا في عندي certain kind of deletions عمالي بتطلع عليهم انا في اكزون معين. For example, exon 19 45 48 51. هذول الاكزونز اللي هم جزء من الجين للدystrophin. فالدystrophin basically is a big gene with many exons. Okay, in this study, the researcher actually investigated 15 unrelated Duchenne muscular dystrophy patients from Saudi Arabia, of course. Okay, and to understand how he made his diagnosis, he actually brought DNA from different individuals, and here is normal. I mean, these are the patients, and this is normal individual who will control, and we have here what's called molecular marker. Okay. هلا الماركر معناها انه في عندنا fragments of DNA we put it in the gel in order to know the molecular weight، يعني قديش الحجم. اوكي. واز criticism، يعني نوع من الانتقاد لهذا النشر لهي البيبر، انه المفروض هو يحط الارقام اللي هون حتى اشوف انا الحجم تبعهم. اوكي. ف it could be it had to be revised by the reviewers لما نشرت هاي الورقه. anyways، هلا في عندي ملاحظات كثير على هذا الموضوع، انه single author for example، وفيش حدا بيعمل هذا الشغل كله لوحده. فسنجل اوثر مينز انه هو راح شخصهم وهو جمع العينات، يعني سحب البلد وهو عمل البي سي ار رياكشن وهو عمل الجل وهو عمل الاناليسيس، وطلعت كل النتائج هاي from one single person which is completely يعني impossible to have this. ولكن بعض هو بس بالدول العربيه بتشوفها هاي، انه مطلوب منك تنشر ورقه بحثيه بس سنجل اوثر، مؤلف واحد. طبعا في لها اسباب كلها شخصيه، انه ليش انا بدي سنجل اوثر، مؤسسه علميه او ابحاث علميه، هذا الحكي مش موجود بالعالم. research is teamwork. anyways، هلا الاندفيدوال رقم 1 و2 و3 و4 هذول على سبيل المثال، هو بحكي لي انه عند individual 1 we have deletion in exon 19. هو كيف عرف انه في عندي deletion in exon 19؟ لما عمل البوليميريز تشين رياكشن، يفترض انه you know this what's the meaning of polymerase chain reaction، بتلاحظ هاي المنطقه هون خاليه من الباند اللي عند النورمال individual. هلا النورمال لازم يكون عنده هذول القطع، اوكي، خاصه بالاكزونز، وبالتالي هو ما عنده هاي الباند، وبالتالي هذا الانديفيدوال عنده ديليشن في هذا الاكزون، وبالتالي اذا في عندي ديليشن الاكزون كبير you will not have the dystrophin as a protein. هلا هذا الانديفيدوال الديليشن جاي عنده for example here I can see it between okay اكزون المفروض هي فوق ال 51 بس انا مش شايف وين هون ديليشن. Here we have 45 deletion in this exon, okay, as well as 48. So we can actually know the type of the exon that is deleted or the area that is deleted in that individual because it's absent, it's not there. Okay, so this kind of disease is related to the absence of the product. You don't have the dystrophin protein. It's not only mutated. We have big deletion mutation ولكن هذا الديليشن ادى انه يكون نفس البروتين is not present or truncated معناها جزء منه فقط اللي صنع. Okay, so you could make a paper from just a case that you could find that is related to certain genetic disorders that we discussed in our course, for example. هلا بنحكي عن بعض glycogen disorders that are related to muscle function. انه لما المسلز بتكون affected. For example, we have what's called McCarthy disease. It's a glycogen storage disease that is related to the glycogen phosphorylase deficiency. هذا الانزيم مش موجود وبالتالي الكولاجين عماله بيترسب بالعضلات او بيتراكم بالعضلات بيؤدي الى المكارتي disease اللي هو affecting the function of muscles and related to other conditions like, for example, hypoglycemia because you are not producing the glucose from these cells. In another way or another example, we have what's called Pompe disease. وما بعرف إذا حكينا عنها في occasion أخرى ولا لا. اللي هو glycogen storage disease. Yeah, I think we talked about this story. We mentioned the movie of Extraordinary Measures. اللي هو هاريسون فورد كان الستار تبعه وهو هاريسون فورد مثل كان البايوكيمست اللي بعدين صار عنده شركه تصنع الانزيمز للتريتمنتس. هلا كانت القصه لهذا الوالد اللي عنده اطفال عمالهم بيعانوا من البومبي ديزيز. بومبي ديزيز في عندهم مشكله هذول الناس في اللايسوسومال انزيمز ما عماله بيكسر الجلايكوجين وبالتالي the glycogen is stored and it will affect muscles actually. عشان كده بتشوفهم على الويل تشيرز هذول individuals. It's autosomal recessive disease and these are the clinical manifestations of the individuals. And you can see here the type of the test that they do in order to diagnose the Pompe disease. It's type 2 collagen storage disease. And just have a look to this movie. It's very interesting from scientific point of view as an example of the genetic disorders that can be treated actually by enzyme replacement. Another condition that is related to the structural formation of the collagen and the skin is the Ehlers-Danlos syndrome, which is associated with different causes like deficiency of the enzymes المسؤولين عن collagen treatments اللي هو البروسيسنج للكولاجين. كيف بصير عمليه بروسيسنج الانزيم بحيث انه يعطي للكولاجين بحيث انه يعطينا right collagen structure. So we could have deficiency in these enzymes or we could have mutations in the collagen types. And as a phenotype, you can see the stretchy skin in these individuals. The osteogenesis imperfecta is another example that is related to the mutation in the genes that are responsible for the collagen synthesis, and we talked about this disease in details. And here is the complete description of the possible mutations that we could have in such kind of disease اللي هم متعلقين بالالفا 1 الفا 2 genes ومتى بيكونوا تايب 1 متى بيكونوا تايب 2 او 3 او 4 ومين منهم dangerous اكثر من الاخر. So you have to be careful when you read this slide as a hint to study smart. And this is the type 2 for example, osteogenesis imperfecta. وبتذكر في احدى المحاضرات كان بالبيوكيمستري اعتقد انه دكتور انت ما حكيت لنا انه في diagnosis. هو مش diagnosis هذا كان مش يعني هو مش بالموديولز كان بالجنرال بيوكيمستري انه هاي diagnostic case. هي مش diagnostic هي example of osteogenesis imperfecta, so you have to recognize it as an example. Okay. هلا when we talk about the function of the muscles, basically we have to talk about the ATP production as well, and this can be applied to the nervous system. which means انه لما بنحكي عن العضلات بنحكي عن الاي تي بي اللي ريكويرد للفانكشن تبع العضلات. اللي حكينا عنه سابقا كان بخص العظام والعضلات متعلق في الاستراكشرز. هلا لما نروح على العضلات شوي بنميل للفانكشن للعضلات والاشياء المتعلقه ب الانرجي برودكشن ان ذا muscle cells. كلنا نعرف انه الانرجي هاوس for ATP production is the mitochondria. So we could have certain muscular disorders that are related to mitochondrial dysfunctions. عشان هيك بنروح بنسميها احنا mitochondrial myopathies، يعني diseases اللي متعلقه بالمسلز ولكن سببها بالmitochondrial. Okay, so as examples that you have to take with you as a take-home assignment, if you would, or to have an idea about these things, we have this list of mitochondrial myopathies that are related to the mutation or deficiency in the mitochondrial function. And most probably, the mitochondrial dysfunction is related to the dysfunction of one of the components that are required for energy production, like electron transport chain, for example. كومبلكسز هذول لانه فيهم بروتينات كثيره، فمتوقع انه اي ميوتيشن ممكن يؤدي الى خلل في الالكترون ترانسبورت تشين، او حتى الكومبلكس نمبر 5 المسؤول عن الاي تي بي سينثيز سينثيز complex. So, for example, we have what's called mitochondrial DNA depletion syndrome as a syndrome of the DNA deletion. And MERF is another condition of the what's called myoclonus epilepsy with ragged fibers. هيك بصير اسم الديزيز. I want you just to have an idea about these disorders. أنا ما رح أسألك عنهم عشان أريحكم بتفاصيل. Just to know that these diseases are related to the mitochondrial deficiency as a function. I mean, not deficiency as structure, but they don't. I mean, the mitochondria doesn't do the function in the right way. So. نكون هيك انهينا الجزئين او المحاضرتين المرتبطين بالبايوكيمستري والاماس السيستم. هلا في اثنين، في رنين رافع ايدها، وفي حدا ثاني. خلينا نشوف بس مين الشباب اللي
معنا. دكتور
رنيم وماجد.
بس بدي أسأل سؤال. بالمحاضرة الأولى حكينا أن الأوستيوكلاست بتعمل أسيديك انفايرمنت عشان مثلا هيدروكسابيبتيت يصير ديجريديشن، وحكينا إنه في ميكانيزم بتعمل فيها بروتكشن لنفسها عشان ما يصير لهاش ديجريديشن بالأسيديك انفايرمنت، بس ما فهمت شو الميكانيزم بتحمي فيها نفسها.
buffering system. Inside she has the buffering system itself.
طب ما بناء على الرسم الموجودة بالمحاضرة البايكربونات اللي هي المفروض تعمل بافرينج تروح على البلاد، ما بتروح مثلا.
احنا مش بنفضيها يا رنين، مش بنفضي البايكربونات. It will be transported in an exchange mechanism with the chloride. So acid base balance is playing a role in this story. You have also carbon anhydrase type 1 which plays a role, and you have buffering system inside the cell. First of all, the bicarbonate buffer. The second thing is the buffering proteins that we have inside the cell. Okay, we have a lot of buffering proteins that work inside the cell. It's not only the bicarbonate. الاسيد نفسه ما بضل بالخليه، ما بضل بالسيتوبلازم. It will be contained in the liposomal large vesicle, if you would, اللي هو الرفل cells or رفل organelle بنسميها، هي مش cell. يعني it's not going to be freely rotating in the cytoplasm. It will be transported actively to the ruffle compartment.
يعني دكتور الاسيديتي هاي بتكون جوا الخليه نفسها، والديجريديشن هيدروكسابيبتيت بيتم داخل
مش
الخليه.
هو الرفل كومبوننت. اعطيك مثال زيها بشبهها تماما. كيف الاميبا بتعمل لايسوسومال فاجوسايتوسيس او الوايت بلاد سيلز بطريقه الاسيد or acidic condition in the lysosomes protecting itself. يعني ليش ما بتقتل حالها باللايسوسومز؟ لأنه it's contained in the lysosomal structure. هلا once we have what's called apoptotic process, by the way، لما يصير في عندنا apoptosis الخلية تبلش تموت، these lysosomal particles will start, or organelles will start to eat the internal organs بالautophagy as well as eating the cell. So we have a regulation process that will keep these vesicles protected from to be mixed with the component of the cell. هي موجودة داخل الفيزيكل داخل الخلية.
يعني degradation بيتم داخل اللايسوزومال بودي الموجود في الاوستيوكلاست.
exact لأنهم جايين على السطح اللي ماسك ب الاوستيويد ستراكشر. ماجد.
تمام دكتور.
هلا
دكتور من بعد إذنك بس هسه أنت حكيت إنه ممكن يكون في يعني لما نعمل للسيرم بيكون في أشياء بتبين معانا للريزوربشن وللفورميشن اوف بون. طب انا كيف بدي اميز هسه بصير فورميشن ولا ريزوربشن بالفحص؟
الكلينيكال اسسمنت ضروري، عشان هيك انا حكيت بالبدايه. هلا في عندك اكس راي، في عندك كلينيكال اسسمنت قائم على انه الشخص من ايش بيعاني. So it could be a tumor, could be bone fracture، هذول بالعاده في bone formation. اوكي هلا ريزوربشن ببين بالاكس راي انه في عندنا shrinkage in the bone structure itself زي like osteoporosis for example. فالاوستيوبروسيس due to the action of osteoclasts is different from the action of other hormones or age-related things اللي هو physiological osteoporosis. فالكلينيكال اسسمنت المبدئي هو بحدد انه what you are going through to diagnose. هلا انت بتشوف الماركرز in general انه they are associated with bone problem، بس بعدين بتقرر بناء على الصور الاشعه اللي عندك، بناء على شكوى المريض نفسه what's going on. يعني for example اذا كان في عنده تيومر بالسباينال بونز you could see these markers.
تمام دكتور. طب ممكن سؤال ثاني دكتور؟
تفضل.
كيف ممكن نشخص إيلرز دانلوس سيندروم؟
هلا as a phenotype stretch scan بالبدايه then you have to analyze the DNA by sequencing. تاخذ عينه بلد تسحب منها الدي ان اي وتعمل سيكونسينج بسيط جدا. يعني we do it here بالجامعه. from research point of view دير بالك يعني مش diagnostic.
لانه في فتاه اجت على قناه المملكه كان معها ايلرز دانلوس سيندروم وراحت على كل دكاتره الاردن ما عرفوا لها، وراحت على كندا ما عرفوا لها، هي شخصت حالها لحالها بعدين صار تدور بالكتب.
والله شوف يا ماجد الان لانه في ريكورد بدي اتحفظ شوي على الكلام. في اشياء بتصير بالدياجنوسيس انا بستغربها وصارت معي شخصيا مع بعض الاشخاص الاقارب انه في مس دياجنوسيس نتيجه عدم القراءه خلينا نقولها. وهذا اللي بحاول انا دائما اثيره في الطلاب. يعني بالمحاضرات انا لا يعنيني انه الطالب يحفظ كل هذول المايوباثيك ديس اوردرز بقدر ما يكون في عنده critical thinking، وهذا بركزوا عليه الان في الطب، والخطه الجديده راح تكون بهذا الاتجاه. الكريتيكال ثينكينج هو اهم شيء، انه انا اشعر بالمريض او احاول اصل لدياجنوسيس صح من خلال معلومات المريض، من خلال الكلينيكال اسسمنت rather than الاكسبيرينس تبعتي القائمه على انه انا قديش شفت حالات وخلاص يلا متعود انا هالتريتمنت مش راح يطلع معي ديزيز. عشان هيك انا بقول للطلاب دائما انتبه انه ممكن تشوف انت certain cases بكره لما تروح ان شاء الله على السنه الرابعه certain cases you could publish يعني many papers from what you will see in the hospital. تمام. فخلي في عندك critical thinking without exaggeration بنفس الوقت، يعني ما اكونش overthinking. الساينس simple يا جماعه. وانا اللي على الجروب على الفيسبوك عندي يوم عملت شير مثلا لواحد من الفيديوز اللي هو ميسلسون اند ستال اكسبيرمنت، كيف السمبل ثينكينج والتواضع عند هذول الاشخاص. از ان ادفايس انا بنصحكم تروحوا تشوفوها، وراح احطها اليوم مره ثانيه على الفيسبوك تبع الالومناي الجروبس. ف في اشكاليات بالدياجنوسيس، وكويس انه راح قرات. يعني انا بعرف شخص ثاني كمان راح قرا وراح شخص حاله وبعدين راح حكى للطبيب انه انا معي كذا كذا. فالمشكله انه ما بيعجبوش يعني، وهي ديروا بالكم انه يعني احيانا you could miss something as a physician مش لانه انت بتعرفش. you could be very busy انك تشوف لك 50 مريض باليوم، برضه هذا لود على الاطباء. يعني احنا ما بنصغر بكتافنا ولا يعني نجلد انفسنا في كونديشنز كثيره. بس انا بركز دائما الطلاب، انا شايف هذا الشيء من 6-7 سنوات الطلاب عندهم بوتنشل للقراءه للريسيرش للكريتيكال ثينكينج. مش رح اقدر اطول اكثر من هيك لانه يمكن حدا في محاضره بعدين ولا لا. هو
لا ما في.
ما في محاضره. انا كثير استمتعت بانه التقيت فيكم شخصا لشخص لما اخذنا بايوكيمستري، والان لسوء الحظ كان بودي انه اعطي المساق او المحاضرات هاي بطريقه مختلفه شوي ونشوف بعض حتى بالمختبرات. فhopefully we can do some research together حتى على مستوى ريفيو. We can meet anytime you want. مكتبي مفتوح، تليفوني مفتوح، الايميل مفتوح. Good luck. Study smart يا جماعه.
شكرا جزيلا دكتور.